In the name of Allah, the most beneficent, the most merciful. Mitochondria is a very important organelle of the cell which provides energy in the form of ATP and called the powerhouse of the cell. Here we show the mitochondria. A mitochondrion contains 1/2 micrometer thick section of chicken brain is viewed with a high voltage electron microscope. When the section is tilted in the microscope, it can be viewed from many different angles, and a large amount of three-dimensional details become apart. Mitochondrion contain inner membrane, outer membrane, matrix, intermembrane space, and cresta, which makes the structure of mitochondria. Membrane individual slices of tomogram are traced. In this case, the inner membrane is traced in light blue, were parallel as outer membrane, and traced in yellow were folded into in cresta that protrude into the mitochondrial interior. The tracing from all the sections are then modeled as three-dimensional surfaces and displaced as three-dimensional model by computer program. In this pyruvate and fatty acids entered the mitochondria and broke down the acetyl CoA. The acetyl CoA is then metabolized by the citric acid cycle, which reduces NAD to NADH. In this process of oxidative phosphorylation, high energy electrons from NADH are then passed along the electron transport chain in the inner membrane to oxygen. This electron transport generates a proton gradient across the inner membrane, which drives the production of ATP by ATP synthase. In combustion, most of the energy would be released as heat if hydrogen were simply burned, but in biological oxidation, by contrast, most of the released energy is stored in the form of useful to the cell by means of an electron transport chain in the inner mitochondrial membrane. As high energy electron is passed along the electron transport chain, some of the energy released drives the three respiratory enzyme complexes that pump hydrogen out of the matrix. As a result, electron chemical proton gradient across the inner membrane drives hydrogen back through the ATP synthase. ATP synthase is an enzyme which makes use of the proton potential created by the electron transport chain in mitochondria. It uses the energy to complete the phosphorylation of ADP to ATP. Pyruvate, inorganic phosphate, and ATP are moved into the matrix while ATP is pumped out. The charge on each of the molecules is indicated for comparison with the membrane potential, which is negative inside. ATP synthase can synthesize ATP by proton motive force or pump proton against their electrochemical gradient by hydrolyzing ATP. ATP synthase can produce when hydrolyzing ATP reveals that synthase can pump.